Hello and welcome to Bio40 Pre-Lecture 29. Time to leave behind the world of molecules and discuss genetic transmission on the population level. We talked before about how mutations are key for evolution, but a mutation only affects an individual. To really understand the relationship between mutations and evolution, we need to observe how mutant and wild-type alleles affect phenotypes and how they are transmitted within populations. Evolution allows us to explain how the diverse and complex life on our planet can arise through strictly natural processes. Throughout history, there have been various models of evolution, but Darwin's version, which is still the basis for our modern understanding, is centered on natural selection. Natural selection requires there to be variations within populations, and these variations must be heritable, at least to some extent. Selective pressures favor individuals with certain variations over others. Such favored individuals are more likely to survive and pass on their traits to their offspring, and the traits become more prevalent with each generation. Darwin was unaware of the concept of the gene as a hereditary unit, but the rediscovery of Mendel's work in the early 20th century led to their ideas being united in what we call the modern evolutionary synthesis. Population genetics helped bring about this synthesis. To discuss population genetics, we'll begin with the Hardy-Weinberg law. This provides a model for how allele frequencies relate to genotype frequencies within a genetically stable population. However, a genetically stable population is not evolving, so we'll see how forces such as selection and mutation change allele frequencies over time. This is known as microevolution. Microevolution may seem insignificant compared to the variation between species, but such large-scale changes, or macroevolution, is simply the accumulation of many smaller events over millions of years. So I used a lot of new terms on that last slide. Let's go over some of them now. Phenotype frequency is the proportion of individuals within a population that have a particular phenotype. Let's say we're looking at a population of 16 people, and 6 of them have the autosomal recessive condition, cystic fibrosis. So the phenotype frequency for the disease is 6 out of 16, or 0.375, while the frequency for the normal phenotype is 10/16, or 0.625. Analogously, genotype frequency is the proportion of individuals within a population that have a particular genotype. While phenotypes can be easily observed, determining genotype often involves molecular analysis, since homozygous dominant individuals and heterozygotes are often indistinguishable. So suppose we learn that 2 of the individuals with the normal phenotype are carriers. That means the genotype frequency for homozygous dominant is 8/16, or 0.5. For heterozygotes, it's 2/16, or 0.125. And finally, the genotype frequency for homozygous recessive is 6/16, which again is 0.375, the same as a phenotype frequency associated with that genotype. Then there's allele frequency, the proportion of gene copies in a population that are of a given allele type. We'll mostly be dealing with diploid populations like ourselves, so that's 2 alleles per individual. The number of copies of a gene within a population will be twice the population number. So for our population of 16 individuals, we have 32 copies of the cystic fibrosis gene. We found there are 8 homozygous dominant individuals. Each has 2 copies of the dominant allele, so that's 16 alleles from that group. Then there are 2 heterozygotes, each with a single dominant allele, adding another 2, for a total of 18. Of course, homozygous recessive individuals will not have any dominant alleles. So that's 18 dominant out of 32 total for an allele frequency of 0.56. You would use a similar approach to find there are 14 recessive alleles, giving a frequency of 0.44. Lastly, let's define this population we keep mentioning. This is simply a group of interbreeding individuals, members of the same species capable of sexual reproduction, sharing the same location at the same time. They share a gene pool, the sum total of all alleles in all individuals within the population. While it's easy to calculate allele frequency from genotype frequency, the reverse is not so simple. If the dominant and recessive alleles each have a frequency of 0.5, then half the population could be homozygous dominant and half homozygous recessive, or the entire population could be heterozygous, or maybe something in between these extremes. The Hardy-Weinberg law helps us determine this relationship as long as a few assumptions hold. These assumptions are that the population is infinitely large, that the individuals within the population mate at random, that there are no mutations and no migration into or out of the population, and that genotypes do not affect an individual's ability to survive and reproduce, so there is no selection. If all of these assumptions are true for a population, we say that population is at Hardy-Weinberg equilibrium. The problem, of course, is that all real-world populations violate at least one, and often many, of these assumptions. This is essentially the equivalent of the spherical cow in physics. The real-life situation is too complex to fully model, so we simplify. And while the simplifications can seem absurd, they can actually get us pretty close to the right answer. If nothing else, Hardy-Weinberg equilibrium provides a null hypothesis we can use in the study of evolutionary forces. The first thing we need to do when calculating Hardy-Weinberg equilibrium is get the allele frequency, which we said is a number of a given allele divided by the total number of alleles in the population. So let's say we have the simple case where there are two alleles for a given gene, a dominant and a recessive allele. These alleles are distributed across homozygous dominant, heterozygous, and homozygous recessive individuals. We need to know how many individuals have each genotype in a given generation. Then we calculate the number of dominant alleles as 2 times the number of homozygous dominant individuals plus the number of heterozygotes. And the number of recessive alleles is the number of heterozygotes plus 2 times the number of homozygous recessive individuals. Then we take the total number of individuals and multiply by 2 for the total number of alleles. Finally, we divide the number of each allele by this total, and we have our allele frequencies. Typically, we call these frequencies P and Q, with P as a dominant and Q as a recessive. These are the allele frequencies for the adults in the population, but their gametes will have the same allele frequencies. We use this information to make a population-level Punnett square. For any random mating within the population, the egg could contribute a dominant or recessive allele, and so could the sperm. But the chance a random egg has a dominant allele is equal to the allele frequency, P. Same with the sperm. So the proportion of homozygous dominant individuals in the next generation will be P squared. Likewise, Q squared will be the genotype frequency for homozygous recessive individuals. Heterozygous offspring can be the result of an egg with a dominant allele being fertilized by a sperm with a recessive allele, or a sperm with a dominant allele fertilizing an egg with a recessive one. The odds of either occurring is P times Q, so the total probability, the genotype frequency for heterozygotes, is 2pq. This is a Hardy-Weinberg equation. At Hardy-Weinberg equilibrium, a population with allele frequencies P and Q will have genotype frequencies of P squared for homozygous dominant, 2pq for heterozygous, and Q squared for homozygous recessive. Note that we can also arrive at this equation algebraically. P plus Q represents all potential eggs, and it also represents all potential sperm. So cross them, or square that quantity, and you get the genotype frequencies of P squared plus 2pq plus Q squared. Using algebra makes it easier to look at more complicated scenarios, such as when a gene has 3 alleles. Take ABO blood groups, for example. The third allele frequency is assigned the variable R. Now all of the sperm and all of the eggs are represented by P plus Q plus R. Square and expand, and you see the genotype frequencies are P squared plus Q squared plus R squared plus 2pq plus 2pr plus 2rq for the 3 homozygotes and 3 heterozygotes, but we'll mostly stick to situations with 2 alleles. There is a specific Hardy-Weinberg equilibrium for each possible combination of allele frequencies. In the most extreme cases, where there's only one allele, of course all individuals are homozygous for that allele. In the scenario where each allele is equally represented, half the population is heterozygous, and a quarter is homozygous for each allele. Note that the frequency of heterozygotes never goes above half, though it is the most common phenotype as long as neither allele frequency is above 2/3. Finally, note that when the allele frequency is high, most of the copies of that allele exist in homozygotes. But when the allele frequency is low, the majority of alleles that do exist are found in heterozygotes. Now let's say we have an initial population that is not at Hardy-Weinberg equilibrium. For an extreme example, imagine a group that's entirely homozygous for one allele, meeting an equal-sized group that is entirely homozygous for a different allele. How long would it take for this newly mixed population to reach equilibrium if the Hardy-Weinberg assumptions hold? Well, with random matings, there's a 50-50 chance each parent is homozygous dominant or homozygous recessive. So a quarter of all matings will have 2 homozygous dominant parents. All of their children will be homozygous dominant, accounting for 1/4 of the offspring of the next generation. Half the matings will have 1 homozygous dominant and 1 homozygous recessive parent, leading to all heterozygous offspring. And the last quarter will involve 2 homozygous recessive parents, and thus homozygous recessive offspring. After just 1 generation of random mating, we're at Hardy-Weinberg equilibrium. So at Hardy-Weinberg equilibrium, the allele frequencies should not change from generation to generation. A single generation of random mating will bring a population to Hardy-Weinberg equilibrium, and it will stay there through subsequent generations as long as the assumptions we talked about aren't violated. However, there is an exception to that second rule. This is when allele frequencies differ between males and females. Suppose all the males in a population are homozygous dominant, and all the females are homozygous recessive. All the offspring will be heterozygous. But then the next generation will have the 1 to 2 to 1 ratio expected at equilibrium. So it just takes 1 extra generation. This is true for an autosomal gene, but it's more complicated when the male and female allele frequencies differ for an X-linked gene. This graph has an example starting with all the women homozygous for a dominant allele and all the men hemizygous for the recessive allele. Note that this means the overall allele frequencies are P equals 2/3 and Q equals 1/3. After 1 generation, the males are all hemizygous for the dominant allele, and the women are all heterozygous. But with each generation, they get closer to converging at a point where the male and female allele frequencies equal each other and the overall frequencies we started with, P equals 2/3 and Q equals 1/3. Because of male hemizygosity, X-linked genes are a little different to think about in general. Having only 1 allele, male allele frequency is the same as genotype frequency. If the dominant allele has a frequency of P equals 0.75, then 75% of males should be hemizygous dominant. But females can still be homozygous or heterozygous. So we can use the Hardy-Weinberg equation just as we do with autosomal genes. Additionally, you have to be careful when calculating allele frequencies for X-linked genes because females have twice as many alleles as males. And that will do it for today. Next time, we start to explore what happens when populations violate the assumptions of Hardy-Weinberg.